In previous video, we already discussed simple carbohydrates, which contain only carbohydrate unit, and it can be branched or unbranched. Now in this video, using our knowledge about simple carbohydrates, we will be discussing about complex carbohydrates. These complex carbohydrates contain carbohydrate unit but with attachment of lipid or protein. If it is attached with protein, then it is called glycoprotein and proteoglycan. And if it is attached with lipid, then it is called glycolipid. But this glycoprotein and proteoglycan, they both are totally different. If the core protein is attached to mucopolysaccharide side chain, which is long, straight and unbranched, then it is called proteoglycan. And if the core protein is attached to oligosaccharide side chain, which is short and unbranched, then it is called glycoprotein. And as discussed in previous video about mucopolysaccharides, it is sulfated and acetylated to hold negative charge. That means proteoglycans are also negatively charged. And we also discussed that this is a cell and this is vascular chamber, and between both of them we have extracellular matrix, and this extracellular matrix contains mucopolysaccharides, which means all the proteoglycans are present in extracellular matrix, and these glycoproteins are present in the remaining two chambers. All proteins present in blood, that is plasma proteins, are glycoproteins except albumin, and all the receptor proteins in the cell are also glycoproteins. This was all about proteoglycans. Now about glycoproteins. They are of three types: O-linked glycoproteins, N-linked glycoproteins, and GPI anchored glycoproteins. Starting with this O-linked glycoproteins. If the carbohydrate side chain is attached to serine and threonine residue of the core protein, then it is called O-linked glycoprotein. And if the carbohydrate side chain is attached to asparagine residue of the core protein, then it is called N-linked glycoprotein. But if the carbohydrate side chain is not attached to any amino acid residue in middle of the protein, and instead it is attached to the carboxy terminal end of the protein, then it is called GPI anchored glycoprotein. So we know that the protein synthesis, that is translation, occurs in rough endoplasmic reticulum. And this rough endoplasmic reticulum, it contains dolicol enzyme, which is necessary for the transport of oligosaccharide side chain to the core protein in case of N-linked glycoproteins. That means this N-linked glycoproteins, their synthesis is co-translational modification, whereas attachment of carbohydrate side chain to serine or threonine does not require any carrier, and it occurs after translation is finished and when the protein reaches the Golgi apparatus. It means O-linked glycoprotein synthesis is a post-translational modification. And in case of N-linked glycoprotein synthesis, this dolicol enzyme, it is inhibited by spermine. Now about GPI anchored glycoproteins, we know that the carbohydrate side chain on one hand is attached to the carboxy terminal end of this core protein, whereas the other hand of this carbohydrate side chain is attached to phosphatidyl inositol of the cell membrane, and the gene which codes for this phosphatidyl inositol is PIGA gene, and because of this attachment it is called glycosylated phosphatidyl inositol anchored glycoprotein. And the examples of this GPI anchored glycoproteins are DAF or CD55 and CD59, and they both inhibit C3 convertase, and this C3 convertase normally gets activated due to acidic pH, especially during night while sleeping due to hypoxia, which forms the membrane attack complex causing RBC membrane to rupture. But in normal person who has this GPI anchored glycoproteins, this DAF or CD55 and CD59 is working properly, which will inhibit this C3 convertase and will stop the membrane attack complex to form. And because of this, our RBCs are safe. But in a person with PIGA gene mutation, there is no GPI anchored glycoprotein, leading to defect in CD55 and CD59. And because of that, there is abnormal activation of complement pathway causing activation of C3 convertase, and therefore membrane attack complex is formed, leading to hemolysis of our RBCs. And as this is occurring whenever the person is sleeping at night, it is called paroxysmal nocturnal hemoglobinuria, which is the only acquired stem cell defect. Thanks for watching. Don't forget to like and subscribe and share with your friends.